properly, then it might actually worsen to a condition called non-alcoholic steatohepatitis that is characterized by inflammation and fibrosis. So you can see that it might not be very clear, but if you guys can see the blue lines, those are basically formation of fibrosis, or basically those are connective tissues. And if this is not properly treated, it can progress to a condition called cirrhosis, where pretty much the whole matrix of the liver has collapsed pretty much. And this might also worsen to carcinoma. So I just want to give a disclaimer that if you guys, for people who love to drink and think that you guys might be able to get a tip or two on how to prevent fatty liver disease, well, I'm sorry to say that the focus of my research will be on the non-alcoholic version. So just to be clear. So why do we care about non-alcoholic fatty liver disease? Well, about 1/3 of the US population has this problem. And about 75% of obese patients have this condition as well. And in fact, almost all morbidly obese patients have this non-alcoholic fatty liver disease. And about 60% of diabetic patients suffer from this condition. So pretty much, it has been clearly shown that this condition is highly correlated with metabolic and cardiovascular diseases. So one of the big problems with this disease is that there's no specific treatment that is specifically targeted to this fatty liver disease. So the most common advice a physician will give you is that, well, lose some weight, that would help you with fatty liver disease. And the other method that's commonly used is type 2 diabetic drug, or metformin. Well, the most common one will be metformin. So what is the cause of triglyceride accumulation? Well, it's really a balance between triglyceride and triglyceride breakdown. So triglyceride synthesis, obviously you're going to get more fat in the liver. Well, if you have less triglyceride breakdown, then there'll be obviously more triglyceride as well in the liver. So we know that, for example, during fasting or starvation, lipolysis will be highly enhanced in the adipose tissue. And that would cause a big influx of fatty acid to the liver, and that would cause an increase in liver triglyceride synthesis. So we also know that high intake or consumption of fructose and carbohydrate can also cause an increase in triglyceride synthesis, and obviously this viral infection called hepatitis can also increase triglyceride synthesis. So the reason why I have this big question mark here is because while a lot of studies have looked at the role of triglyceride synthesis in causing hepatic triglyceride accumulation or hepatic steatosis, very little is known about the role of triglyceride breakdown. So what do we know about lipolysis or triglyceride breakdown in the liver? So for the longest time, prior to 2004, it was known, or it was thought that this lipase or enzyme called hormone-sensitive lipase, HSL, was thought to be the main triglyceride lipase that breaks down triglyceride. So triglyceride really is just a glycerol backbone, which is water-soluble, attached to 3 different fatty acids. And what hormone-sensitive lipase was thought to do, and is still thought to do, is to break down triglyceride and basically cleave off a fatty acid and release a fatty acid and also the acylglycerol, which has 2 fatty acids left. So besides being able to break down triglyceride, hormone-sensitive lipase can also break down the acylglycerol. So it has activity towards both triglyceride and the acylglycerol. So in 2002 or 2003, a group basically generated a hormone-sensitive mice. So what they did was they basically knocked out this enzyme or lipase in the mice. So the mice completely lacked this lipase. And then they look at basically different lipid species. And to their surprise, they did not see a triglyceride accumulation in white adipose tissue or brown adipose tissue where this enzyme is highly expressed. So there was really no difference in the amount of triglyceride in the liver. But then what they saw was there's this increase in the amount of diacylglycerol of these 2 different diacylglycerol species, suggesting that perhaps hormone-sensitive lipase is a lipase predominantly for diacylglycerol and not so much for triglyceride. So the question mark then is, well, what might then be the main or predominant lipase to triglyceride? So again, question mark. So in 2004, 3 different labs actually discovered this lipase or enzyme called adipose triglyceride lipase (ATGL) in adipose tissue where it's highly expressed. So it was known that ATGL is highly expressed in adipose tissue, including white and brown adipose tissue, and not so much in the liver or in the heart or in the muscle. And when they generated a whole body knockout of this ATGL, they actually saw fat accumulation not just in adipose tissue but also in multiple, well, basically ectopic triglyceride accumulation. So basically they saw accumulation of fat and triglyceride in the liver, in the muscle, and it's also in the heart. As I will be talking about. So this one thing about this knockout model or knockout mice is that they actually die within about 14 to 16 weeks of age due to the fact that they accumulate so much fat in the heart. So basically, you can see this figure. So the ATGL knockout mice, they accumulate so much fat in the heart that you can see it's way bigger and it's obviously way yellowish compared to the wild type. So they basically die from heart failure. So another phenotype that was observed is that these ATGL knockout mice also exhibit a lower lipolytic activity in the liver. So basically, there was just lower lipolysis in the liver. So this may suggest that, well, although ATGL may be expressed pretty low, I mean, relative to adipose tissue, it might still play a major role in lipid metabolism. So we were really interested in the role of ATGL in the liver, specifically in the liver. So we know that liver is an important organ in terms of detoxifying, xenobiotics or poisons or toxins. But then besides that, it's also an important organ in terms of making lipoprotein that is important in transporting all kinds of different, many triglyceride cholesterol to the whole body. So we really wanted to investigate if hepatic ATGL plays an important role in lipid metabolism and also whole body energy metabolism. So as I mentioned before, insulin resistance and glucose intolerance is also highly associated with non-alcoholic fatty liver disease. So we wanted to investigate as well if hepatic ATGL might play a role in the development of insulin resistance. And the last objective of my research is to basically elucidate the mechanism through which hepatic ATGL may mediate a pathway that is downstream of ATGL. So basically ATGL has been shown to play a role in triglyceride breakdown, but how about pathway that is below that, for example fatty acid oxidation? So we'll be looking into that shortly. So in order to study the role of hepatic ATGL, we decided to use adenovirus. So basically it's a method that is specifically targeted to the liver. So what we did was we used adenovirus that encodes either a control or ATGL shRNA. So the ATGL shRNA is going to suppress the expression of ATGL, and because adenovirus, so it's going to be specific to the liver. So as you can see here, we got a very good knockdown, about 70% at the gene expression level, and when we did and we showed that ATGL protein expression was also decreased by about 60%. So we also performed a triglyceride hydrolase activity, and we got about 40% knockdown, and this is less because there are other as well for triglyceride in the liver. So one of the things, I probably should mention this. So what we did was we injected the adenovirus with encoding this shRNA and via tail vein, and then after switch the diet to either a control or a child diet or high-fat diet, and let them go and basically harvest the tissues and serum 7 days after the injection. So really they were only on the child or high-fat diet for 7 days, so it's really a short-term high-fat diet. And we basically fasted them overnight and then the tissues and serum. So one of the big phenotype that we saw is that the ATGL knockdown adenovirus causes increase in liver weight regardless of diet type. So we are curious if this is due to the effect of decreased triglyceride hydrolase activity. So we look at liver triglyceride, and again, this data basically support what we saw in the liver weight data. So liver triglyceride was increased by at least twofold, both in the child and high-fat diet fat mice. So this is basically just a liver section, and you can see that in the control, you can barely see any lipid droplets, but then as depicted by the arrows, you can see this white circle globule of fat that clearly is way more than in the control. So ATGL knockdown causes this condition called steatosis or fatty liver disease. So now that we have shown ATGL is involved in triglyceride breakdown, we wanted to see what is downstream of ATGL. So we show that ATGL can break down triglyceride to fatty acid and diacylglycerol. And we know that the fate of fatty acid in the liver can go either two ways. So one of them is it could be incorporated back into triglyceride, where this triglyceride can be repackaged into VLDL, very light density lipoprotein, as a way to transport triglyceride to the other tissues or organs. Or the fatty acid can actually be burned in the mitochondria, so basically can undergo beta oxidation as a source of energy. So in order to characterize what happens to that is being hydrolyzed by ATGL, we first triglyceride, so basically blood triglyceride. And as you can see here, ATGL knockdown basically had no effect on serum triglyceride. So we wanted to specifically look at VLDL secretion. So as I mentioned before, fatty acid could be made into triglyceride and then repackaged into VLDL. So we wanted to look at specifically VLDL secretion. So we use thiloxapol, which basically inhibits lipoprotein lipase. So lipoprotein lipase basically is involved in the clearance of serum of VLDL. So by inhibiting this, we would be able to measure basically VLDL synthesis and secretion. So as you can see, ATGL knockdown had no effect on serum triglyceride or even VLDL synthesis and secretion. So we are like, okay, maybe it would have an effect on beta oxidation, which is the other potential route of fatty acid. So what we did here was we basically isolated primary hepatocytes, so actually Mara did, and what we did was we did this pulse or chase experiment where we added radiolabeled oleate. So this is basically a tracer experiment to look at where does the oleate, where would this oleate go at the end. So basically we added radiolabeled oleate for 2 hours to the cells. And after 2 hours, this is the part where we actually incorporate this oleate mostly into triglyceride fraction. And then after 2 hours, we basically harvest the cells and also replace the media for another set of cells, so similarly treated. And then let it go for 6 hours. This is where we actually look at how much of the incorporated oleate into the triglyceride will get broken down into fatty acid and then being burned in beta oxidation. And then we harvested both the media and cells. So what we did here was we did this ATGL knockdown experiment represented by the red bar, and again consistent with the serum triglyceride data in vivo in mice. We saw no change in media triglyceride or basically VLDL secretion. However, when we knock down ATGL, we look at beta oxidation, well specifically look at ASM, which is a good measure of beta oxidation. We saw this decrease in beta oxidation, so kind of suggesting that, well, maybe ATGL actually is more involved in beta oxidation and not VLDL secretion. So we did the opposite here where we overexpressed ATGL, and again there was no effect on media triglyceride or VLDL secretion. With the ASM we saw this induction, so more than 3-fold increase in beta oxidation as a result of overexpressing ATGL. So basically these data match each other pretty well, and suggesting that ATGL partitions the fatty acid that it hydrolyzes to beta oxidation or oxidative pathways and not to VLDL synthesis. So now that we have shown that ATGL preferentially channels fatty acid to beta oxidation where it gets burned as a source of energy, we wanted to kind of investigate what is a transcriptional mechanism or what is a transcriptional regulation that may regulate the effect of ATGL on beta oxidation. So obviously we went to look at PPAR alpha. So PPAR alpha is the main nuclear receptor that regulates genes that are involved in beta oxidation. So for example, during fasting, PPAR alpha is usually upregulated because during fasting we need to burn fats. This is one of the ways that we increase beta oxidation is via upregulation of this PPAR alpha expression. So PPAR alpha is obviously located in the nucleus. And what is known about this nuclear receptor is that it can be activated by fatty acid. So our initial hypothesis is that perhaps ATGL breaks down triglyceride, releasing fatty acid, and that fatty acid may be able to bind to PPAR alpha in order to activate fatty acid oxidation. So we wanted to look at if that actually happens. So what we did was we basically looked at gene expression of PPAR alpha and some of the other genes that it actually regulates. And you can see that regardless of diet type, ATGL knockdown causes about 60 to 70% knockdown regardless of diet type, suggesting that ATGL regulates PPAR alpha. So the objective, the summary for the first objective is that we have shown that ATGL is a major hepatic triglyceride lipase that breaks down triglyceride into fatty acid. And we also show that fatty acid, this fatty acid is preferentially channeled to beta oxidation in the mitochondria and not towards VLDL secretion, where it could be secreted as a form of serum triglyceride. So we also showed that ATGL basically regulates PPAR alpha. However, the mechanism is not clear yet. So maybe it's not, I didn't show that it's definitely the fatty acid, but I did show that ATGL somehow regulates PPAR alpha, as you can see in the previous figure. So moving on to the second objective, I wanted to investigate whether, well, I showed that ATGL knockdown causes fatty liver disease or triglyceride accumulation in the liver. So I wanted to investigate if ATGL also contributes to the development of insulin resistance and glucose intolerance that is highly correlated with fatty liver disease. So this is basically a figure showing basically comparing obese, overweight children. So basically comparing overweight children with normal hepatic triglyceride level and those that have higher or more than 5% fat in the liver. So you can see that these children actually have less insulin sensitivity, suggesting that they are more insulin resistant because they have more fat in the liver. So it is well known that fatty liver disease and insulin sensitivity decrease is highly correlated. We don't know exactly which comes first, it's a chicken and egg story, but then we know that with decreased insulin sensitivity, the liver is highly affected in the sense that usually when there's insulin as a signal, hepatic glucose production would be decreased, but because of decreased insulin sensitivity or increased insulin resistance, the liver keeps cranking up more glucose, although it's not supposed to when it's signaled by the insulin. And the other big observation or hallmark is that the glucose uptake in skeletal muscle and adipose tissue is also reduced because the role of insulin is obviously to stimulate glucose uptake into this muscle and adipose tissue. So these two conditions would then lead to obviously high blood glucose level. So what we did here was basically just to look at serum glucose level of mice that lack this ATGL in the liver. And as you can see, despite accumulating more fat in the liver, we were surprised to find that ATGL knockdown actually causes a decrease in serum glucose level. So blood glucose level was actually decreased, suggesting that maybe they are actually more glucose tolerant despite the fact that they accumulate more fat in the liver. So if you look at the insulin level, while there was no effect when the mice were fed the chow diet with ATGL knockdown, in the high-fat diet, you can see that ATGL knockdown actually abrogate, this is not a single increase, but basically we see a decrease in serum insulin level as a result of ATGL knockdown, suggesting that they perhaps might be actually more insulin sensitive too, despite the fact that they have more fat in the liver. So this is basically glucose tolerance test. So what we did here was we basically gave a bolus of glucose to the mice, and then we monitored the blood glucose level over minutes. And as you would expect, when you give a glucose load, there's this big increase in blood glucose level. But then as you can see, as represented by the red line, the ATGL knockdown mice actually were able to clear the glucose level much faster than the control mice. Again, suggesting that although they have more fat in the liver, they seem to be more glucose tolerant, so they were able to clear the glucose level faster. So we were interested to look at what happened to the insulin signaling. So usually in insulin resistant, insulin signaling is defective. So we look at two common markers of insulin signaling, IRS1 and AKT, and we look at phosphorylation as a way to measure insulin signaling. And as you can see here, if you compare the two white bars, those are basically control and ATGL knockdown under no insulin. And we don't see an impact insulin signaling. And the good news as well is that ATGL knockdown mice were still able to respond to insulin, suggesting that hepatic insulin signaling was not affected in these mice lacking ATGL. So now that we have seen that ATGL knockdown were able to rescue or normalize glucose intolerance, we were wondering if this hepatic ATGL knockdown will also have an effect on obese mice that were diet induced. So basically, what we did was we feed high-fat diet to these mice for 3 months to cause obesity, to cause insulin resistance and obviously also fatty liver. And we wanted to see if ATGL knockdown would be able to rescue that phenotype. So again here, basically I look at gene expression of ATGL and protein expression of ATGL as a result of the adenovirus, and again we got a very good knockdown at both mRNA and the protein level. So we also look at body weight, and as expected, high-fat diet for 3 months caused this increase in body weight. What we saw was ATGL knockdown actually slightly decreased body weight as well. So that was pretty exciting. So we again look at liver weight, which is what we are really interested in. And as you can see, ATGL knockdown again causes increase in liver weight. And obviously this data again is supported by the liver triglyceride data. So as you can see here, ATGL knockdown causes increase in liver triglyceride as shown before. And then with high-fat diet, again, it causes accumulation of triglyceride. But then when we knock down ATGL in the liver and also feed them with high-fat diet for 3 months, it actually further increases the amount of triglyceride in the liver, suggesting that they are very, very steatotic. So this is really interesting because we really want to look at if ATGL knockdown can still rescue this glucose intolerance caused by diet-induced obesity. So what we did here was we again looked at serum glucose. So this is under fasting, so overnight fasting. Just take note. And again here, ATGL knockdown again decreased serum glucose level. And the best part is that ATGL knockdown also basically abolished the increase in serum glucose level caused by high-fat diet. So here we look at serum insulin, and again this is very, which is what I wanted to see, and you can see that ATGL knockdown basically again abrogate the increase in serum insulin level caused by high-fat diet. So suggesting that ATGL knockdown is still able to rescue the serum glucose intolerance phenotype in these diet-induced mice. So here again we did the oral glucose tolerance test, and what we did again was just to give a bolus of glucose and then monitor the blood glucose over time. And if you look at the line, the very top represented by the white triangle, you can see that with the high-fat diet it causes this big spike in serum glucose level. But then when you knock down ATGL, represented by the red triangle bar, basically the serum glucose level was being able to clear very much faster, almost completely recovered to the basal level. So suggesting that again these mice are actually very glucose tolerant, so they were able to clear the blood glucose level much faster when they lack ATGL, although they have fatty liver disease. So this is really a model that kind of uncouple or kind of dissociate what has been observed frequently. So we have previously shown that ATGL, when we knock down ATGL, we get decrease in fatty acid oxidation. So what we did here was we really isolated mitochondria and then look at fatty acid oxidation specific mitochondria. So while ATGL knockdown had no effect on mitochondrial fatty acid oxidation under controlled diet, you can see that it actually rescued the increase in fatty acid, not rescue, but basically decreased the fatty acid oxidation in the high-fat diet mice, suggesting that again that ATGL channels preferentially to beta oxidation. So the next thing we were like, well, if fatty acid oxidation is defective, what might actually occur to the other common substrate pathway, which is glucose oxidation or glucose utilization? So what we did here was we look at liver glycogen. So under high-fat diet, you can see that ATGL knockdown caused a decrease in liver glycogen. So glycogen basically, as you guys probably know, is the storage form of glucose in the liver and also in the muscle. So when we see this decrease, we're like, well, perhaps maybe there's an increase in glucose utilization as a result of defective fatty acid oxidation. So to confirm that, we isolated primary hepatocytes, and then we look at glucose oxidation. And as you can see here, ATGL knockdown causes increase in glucose oxidation in the hepatocytes, suggesting that perhaps as a result of defect in fatty acid oxidation, these hepatocytes lacking ATGL compensate by increasing glucose oxidation as a source of energy. So besides glucose oxidation, we wanted to see if maybe glucose production in the liver might also have an effect on the improved glucose tolerance that we saw in the ATGL knockdown mice. So what we did here was we again look at glucose production in primary hepatocytes, and you can see that there was this decrease in glucose production. So we also wanted to look at some of the other gluconeogenic genes that are involved in glucose production. And again, what we expected to see is that there's this decrease in these genes that are involved in glucose production. So to really look at glucose production in vivo, we decided to perform pyruvate tolerance test. So what we did here was we basically gave pyruvate to the mice and then monitor the blood glucose level. So pyruvate is a substrate for glucose production. So over time, over about 90 minutes, you can see that ATGR knockdown basically have a lower serum glucose level, suggesting that they had lower glucose production in the liver. So when a mice, so when ATGL is normally expressed, fatty acid oxidation, glucose oxidation, and also glucose production are basically in homeostasis. But then, when we knock down ATGL, we saw an increase in lipid accumulation in the liver, specifically triglyceride. And what happened is that we also saw a decrease in fatty acid oxidation. So as a result of that, to compensate for the lack of energy source, we basically saw an increase in glucose oxidation and also a decrease in glucose synthesis, suggesting, and one of the big observation that we saw is also that ATGL basically knocked down, although it caused hepatic triglyceride accumulation, it did not impair insulin signaling. So suggesting that ATGL actually uncouples hepatic triglyceride accumulation from insulin resistance and glucose intolerance. So next we wanted to investigate the mechanism of how ATGL mediates beta oxidation and also PPAR alpha activation. So we wanted to investigate the role of a fatty acid binding protein that is involved in fatty acid transport. So, which is liver fatty acid binding protein, LFABP or FABP1. So fatty acid binding protein is basically the most abundant cytosolic protein in the liver. I think that's about 3 to 5% of, it basically comprises 3 to 5% of all the cytosolic protein in the liver. So it is highly expressed in the liver. Some can be found as well in small intestine and in the kidney. I'll go further into that. But then there are also other isoforms of FABP. So it has been shown that LFABP is involved in carrying the fatty acid to the mitochondria to facilitate beta oxidation. It has also been shown that LFABP can carry fatty acid to the nucleus to basically deliver fatty acid or a ligand to the PPAR alpha. So LFABP has also been shown to interact directly. So LFABP has also been shown to interact directly with PPAR alpha to modulate its activity and also some of the other target genes. So basically what we wanted to look at is, well, LFABP has been shown to carry fatty acid to both mitochondria and also to the nucleus to assist with beta oxidation and also PPAR alpha activity. So we wanted to look at if FABP is also involved in channeling specifically ATGL hydrolyzed fatty acid to the mitochondria for beta oxidation or to the nucleus to activate PPAR alpha. So what we did was we decided to use wild-type black 6 mice or LFABP knockout mice. So basically this is a mice that lack LFABP, it's a whole body, so it doesn't have LFABP. So what we did here was we basically injected ATGL shRNA adenovirus through the tail vein, and then 7 days later we harvested the serum and tissues after overnight fast. So as you can see here, well, this has been knockdown causes increased liver weight, and in the FABP knockout we still saw an increase in liver weight, and this is also supported by the liver triglyceride data where ATGL knockdown still caused an increase in liver triglyceride, although you can see that the increase is slightly less. So surprisingly, although ATGL knockdown is involved in triglyceride breakdown, which releases fatty acid, we didn't see an effect on liver fatty acid. As you can see, ATGL knockdown basically didn't change the amount of fatty acid in the liver, regardless of genotype. So this is where the data gets really interesting. Is where we basically overexpress ATGL basically in primary hepatocytes. So again, we did a Powell's or Chase experiment where we look at how much of the OLE is incorporated into triglyceride and how much of the triglyceride gets broken down to fatty acid and then being burned in as ASM or beta oxidation. So as you can see, this is the triglyceride incorporation during the 2 hours of Powell's. And you can see that ATGL, when you overexpress ATGL, there's attenuated incorporation of radiolabeled OLE to the triglyceride fraction, which is expected regardless of the genotype. And we then look at basically ASM or beta oxidation during the chase period, where we really look at how much of the incorporated triglyceride is being broken down into fatty acid. And you can see that with the ATGL overexpression, we get this big induction in fatty acid oxidation. And then when we look at primary hepatocytes isolated from FABP knockout mice, there was a decrease in the beta oxidation, as has been shown previously. But then when we overexpress ATGL, we still see an increase in beta oxidation despite the fact that LFABP is absent. So suggesting here that LFABP perhaps is not important in mediating ATGL regulated beta oxidation. So perhaps LFABP doesn't actually carry ATGL hydrolyzed fatty acid to the mitochondria for beta oxidation. So like I mentioned previously, there are other isoforms of FABP. So what I was really looking at is the liver fatty acid binding protein because it's the most predominant one in the liver. But then there are some other FABP as well, like intestine FABP, heart FABP, keratinocyte FABP, adipose FABP, and brain FABP. So we wanted to discount, we wanted to look at if other FABP isoforms might play a role as well in ATGL-mediated beta oxidation. So what we did was, instead of using an LFABP knockout mice, we used an inhibitor called HTS that basically inhibits all the FABP isoforms. So not just the LFABP, but all of the other 5 isoforms as well. So again, here what happened is when you overexpress ATGL, there's less incorporation, supporting what we just showed in the previous figure. And then when we look at chase beta oxidation, again there's this induction in oxidation as a result of overexpressing ATGL. But then when we added this inhibitor to all the FABP isoforms, we still see an increase in beta oxidation when we overexpress ATGL. So again, here suggesting that, well, it seems like none of the FABP seems to mediate the role of ATGL on channeling fatty acid to beta oxidation. So now that we have shown so now that we have shown FABP is not involved in channeling fatty acid hydrolyzed by ATGL to the mitochondria, we were wondering is, well, might it play a role in channeling fatty acid from ATGL to PPAR alpha to activate the genes? So here again we look at the expression of PPAR alpha and some of the other target genes that are involved in fatty acid oxidation or beta oxidation. And again here, when you overexpress ATGL in the wild type, of course we saw this increase. But then again, with the FABP knockout, we still see this upregulation of most of the genes despite LFABP being absent. So again here suggesting that, well, it seems like LFABP may not be involved in either channeling ATGL hydrolyzed fatty acid to the mitochondria for beta oxidation or even to the nucleus for activation of PPAR alpha. So that was fun. So now that we have discovered the role of LFABP in channeling fatty acid as a ligand to activate PPAR alpha, we were wondering, well, maybe ATGL actually regulates